Introduction. Welcome, medical students, to today's lecture on the intricate mechanisms underlying dystrophin protein deficiency. In this comprehensive script, we will explore the various aspects related to this condition, including the molecular pathways involved, the role of specific proteins, the impact on cellular processes, and potential therapeutic strategies. Let us dive into the fascinating world of dystrophin protein deficiency and unravel its complexities. Dystrophin protein deficiency, DPD, is primarily caused by mutations in the dystrophin gene located on the X chromosome. This gene encodes a crucial cytoskeletal protein called dystrophin, which is responsible for maintaining the structural integrity of muscle fibers. Without functional dystrophin, muscle cells become susceptible to damage and degeneration. The absence or insufficient expression of dystrophin leads to the development of Duchenne muscular dystrophy, DMD, or its milder form, Becker muscular dystrophy, BMD. The specific mechanisms underlying DPD involve the disruption of important cellular processes, including the dystrophin-associated glycoprotein complex, DAGC, calcium homeostasis, inflammation, mitochondrial dysfunction, and epigenetic modifications. The dystrophin-associated glycoprotein complex, DAGC, plays a crucial role in maintaining the structural integrity of the muscle sarcolemma. This complex consists of several integral and peripheral membrane proteins, including dystrophin, dystroglycans, sarcoglycans, and syntrophins. These proteins ensure the connection between the cytoskeleton and the extracellular matrix, facilitating muscle contraction and providing necessary stability. Disruptions in the DAG result in increased susceptibility to membrane damage and subsequent muscle degeneration. Calcium plays a fundamental role in muscle contraction, and its regulation is crucial for proper muscle function. DPD disrupts calcium homeostasis by impairing the influx and efflux of calcium ions across the sarcolemma. This dysregulation, coupled with the compromised structural integrity of the membrane due to dystrophin deficiency, leads to increased intracellular calcium levels, triggering detrimental effects on muscle cells. These perturbations contribute to cellular toxicity, oxidative stress, activation of proteolytic pathways, and subsequent muscle fiber degeneration. Inflammation plays a critical role in the pathogenesis of DMD. The absence of functional dystrophin triggers the release of pro-inflammatory factors, such as cytokines and chemokines, leading to a chronic inflammatory response in affected muscles. Inflammatory mediators, including tumor necrosis factor alpha, TNF-alpha, interleukin-1 beta, IL-1 beta, and interleukin-6, IL-6, further exacerbate muscle damage by promoting oxidative stress, increasing proteolysis, and impairing muscle repair mechanisms. These inflammatory processes contribute to progressive muscle degeneration and the clinical manifestations observed in DMD patients. Mitochondrial dysfunction has emerged as a significant contributing factor in DMD pathogenesis. The absence of dystrophin affects mitochondrial function by disrupting calcium handling, impairing energy metabolism, and initiating oxidative stress. These alterations lead to mitochondrial damage, reduced ATP production, and increased production of reactive oxygen species, ROS, ultimately culminating in muscle cell death and degeneration. Biomarkers play a vital role in the diagnosis, monitoring, and assessment of therapeutic interventions in DMD. Various biomarkers have been identified, including creatine kinase, CK, lactate dehydrogenase, LDH, and cardiac troponin levels. These biomarkers reflect muscle damage, inflammation, and cardiac involvement. Additionally, advances in molecular profiling techniques have enabled the identification of gene expression signatures and microRNAs that offer insights into disease progression and potential therapeutic targets. Myofiber remodeling is a complex process implicated in the pathogenesis of DMD. Loss of dystrophin leads to muscle fiber necrosis, followed by cycles of regeneration and fibrosis, ultimately resulting in the replacement of healthy muscle tissue with non-functional fibrotic scar tissue. Activated fibroblasts, excessive collagen deposition, and dysregulation of extracellular matrix components contribute to impaired muscle function, contractility, and progressive muscle weakness. Epigenetic modifications have gained significant attention in DMD research. These alterations, including DNA methylation, histone modifications, and microRNA dysregulation, contribute to the aberrant gene expression observed in DMD patients. Epigenetic changes affect key biological processes, such as muscle development, inflammation, and fibrosis. Understanding these modifications is vital for elucidating disease mechanisms and developing targeted therapeutic strategies. Emerging therapeutic approaches in DMD focus on modulating the immune response to attenuate inflammation and subsequent muscle damage. Immune-based therapies, such as corticosteroids and immunomodulatory agents, aim to reduce pro-inflammatory mediators, promote muscle regeneration, and delay disease progression. These treatments have shown promising results in improving muscle function and slowing the decline in DMD patients. RNA editing and splicing represent innovative avenues for therapeutic intervention in DMD. This approach involves modifying or correcting RNA transcripts to restore the correct reading frame and produce functional dystrophin proteins. Techniques such as exon skipping, antisense oligonucleotides, and CRISPR/Cas9 gene editing offer potential strategies to overcome the underlying genetic defect in DMD patients. Satellite cells are crucial for muscle regeneration and repair. These resident stem cells contribute to muscle growth and repair in response to injury or stress. In DMD, satellite cells exhibit dysregulation and impaired function due to the absence of dystrophin. Understanding the intricate signaling pathways governing satellite cell activation, proliferation, and differentiation may pave the way for novel therapeutic approaches to enhance muscle regeneration in DMD patients. Conducting preclinical studies and well-designed clinical trials is essential for the development of effective therapies for DMD. Preclinical models, including animal models and cell culture systems, provide valuable insights into disease mechanisms and therapeutic targets. Clinical trial design involves careful consideration of trial endpoints, patient population, outcome measures, and safety evaluations. These studies play a crucial role in assessing the efficacy, safety, and long-term effects of potential treatments. In conclusion, dystrophin protein deficiency encompasses a diverse range of complex mechanisms that contribute to the pathogenesis of DMD. Understanding these mechanisms, from the disruption of the dystrophin-associated glycoprotein complex to the involvement of calcium homeostasis, inflammation, mitochondrial dysfunction, and epigenetic modifications, is crucial for developing targeted therapeutic strategies. With ongoing advancements in research, we hope to unlock new treatment avenues and improve the quality of life for individuals affected by DMD.